respected chairperson, distinguished guests, esteemed faculty members, researchers, students, and participants. A very warm good evening to everyone. It is my great pleasure to introduce our esteemed speaker, Dr. Inderpal Kaur, biochemist, Regional Research Station PAU, Kapurthala. Dr. Gaur completed her PhD in biochemistry from PAU, Ludhiana, with specialization in biochemistry and biotechnology. She has previously served as a research fellow at PAU at Guru Angad Dev Veterinary and Animal Science University, Ludhiana, where she worked on several prestigious research projects in molecular biology, virology, and biotechnology. She has published numerous international research papers, book chapters, and several popular scientific articles. Her research focuses on plant biochemistry, molecular biology, biotechnology, and crop improvement. She has also been recognized with the ICAR ASRB NET qualifications, university merit scholarships, and a fully funded international scholarships from the government of Israel to attend the Summer Institute on Food Safety and Security at Tel Aviv University. With her rich experience in teaching, research, and innovation, Dr. Inderpal Kaur continues to inspire students and researchers through her valuable scientific contributions. Today, we are privileged to have her with us to share her knowledge and expertise. On behalf of Edu-Mentors Pvt Limited, I extend a heartfelt welcome to Dr. Inderpal Kaur and thank her for accepting our invitation. Ladies and gentlemen, please join me in giving a warm virtual round of applause to welcome our distinguished speaker, Dr. Inderpal Kaur.
Thank you so much for my introduction. Thank you so much. So, good evening to all present here. Okay. So, the topic of the discussion for today is the role of biotechnology in creating sustainable agriculture and livestock. Okay. So there is a brief introduction about the sustainable. There are 17 sustainable development goals, in which the very first is no poverty, and the second one is zero hunger, and the third is good health and well-being. So our as an agriculturalist, our main goal is for zero hunger and no poverty. That ultimately leads to good health and well-being. So, as our topic is role of biotechnology in creating sustainable agriculture and livestock. So, what do you understand from the term sustainable? Sustainable word is derived from sustain, which means to maintain, uphold, and endure. Sustainable agriculture development attempt to meet society's food and fiber demand with long-term solutions for current and future generation. That ultimately lies on the second goal, that is a zero hunger. So the primary requirement for sustainable agriculture is to merge a healthy environment and economic profitability with social and economic equality, and to develop climate resilient agriculture and livestock, so that we can attain the first goal, no poverty. So, to attain this goal, sustainable agriculture scientists must use multidisciplinary approaches that combine biology, engineering, chemistry, economics, community development, so that we can achieve one first or second goal. So, as we all know, that in modern era, population increases, so the land requirement also increases for residents and for other infrastructure facilities. Therefore, ultimately the agricultural land get reduced, but the demand of population increases. That means we have to maintain or use the limited resources and the produce that is sufficient for food to achieve food safety. So this picture represents that from 1940 to till date, if the requirement of the grain is same, but the land area, it reduces. Therefore, the modern technologies have to intervene the agriculture practices to produce more yield in less area, so that in 120 meter square plot we can get approx 250 kg of yield, though we were achieved it in 1940 in 1347 meter square area. So here the biotechnology or the other modern technologies are required, so that we get the same produce but with limited inputs or with resource efficiency use. So the biotechnology can be used to create crops that are resistant to pests and diseases to address these issues. So the the soil nutrient deficiency, it is also a main obstacle that to in this sustainable agriculture, because by this soil nutrient deficiency, the it ultimately reduces the crop yield and plant health. So biotechnology has the potential I'm so sorry for the inconvenience. I'm so sorry. Extremely sorry for this fault. Okay. where I was at the third slide. Am I audible?
Yes, ma'am.
Okay. I'm so sorry. Okay. So, as I introduced the sustainable goals, so our target is for sustainable agriculture. That means we have to uh, fight against goal number 1 and 2, that is zero hunger and no poverty. So, in this slide, if we can understand the this from the year 1940 onwards. So, as in modern era, the as population increases, so the land requirement also increases for residents and for other infrastructure facilities. Therefore, the agriculture land reduces, but the demand of population increases. Why? Because everyone need food. But we have to maintain, or we can say use the limited resources to produce sufficient amount of food to achieve food safety goal. So this picture represents that from 1940 to till date, if the requirement of grain is same, but the land area reduces. Therefore, the modern technologies should be intervened in traditional agriculture practices to produce more yield in less area. For example, in this picture, in nowadays, if we have only 120 meter square area, the grain yield should be the 250, as in 1940, where 1347 meter square area was used for the cultivation of 250 kg. Now, what is the difference between conventional agriculture and sustainable agriculture? So, conventional agriculture, the first point is the chemical inputs. In conventional way, we use a lot of chemicals that ultimately reduces biodiversity, and due to the chemical intervene, the economically it's not favorable, and chemicals also emits greenhouse gases that affects environment, and due to the effective cost of this the chemicals, the great socio-economic gap get there is a obstacle for this. But why we want to achieve this agriculture sustainable agriculture goal, to first of all to reduce the socio-economic gap. So for this we have to eliminate or reduce the chemical input by mediating the microbes, plants, and plant and their derivatives such as plant based biopesticides, that ultimately preserves biodiversity and enhance economic stability, and ultimately reduces the greenhouse gases, so environment must be safe, and our socio-economic gap it also get reduced. So what does it need to be sustainable? Why this goal is required? What is the reason behind that? To attain economic stability, to attain diversification, and to attain the environmental mentally safe and sound area for living. The third one is for the social values, for healthy and good families and communities. We have to be sustainable so that we can get economical, social, or cultural benefit. And the fourth one is for food and fiber security. That ultimately we have to increase our productivity and to improve the resilience. So, how biotechnology helped in this case? Uh, Biotechnology is an alternative method to the current approaches, that is, the traditional approaches to improve the environmental, livestock, and agriculture system. So, it helps to reduce fertilizers and pesticides application in the current agriculture production system that ultimately affects environment and our health also. It is also an effective strategical approach to develop different high yielding and stress tolerance crop varieties so that we can get a sufficient amount of yield in less area. So, what is the basis of it? How it works? it works on expression. As we all know, there is a central dogma that is from DNA to RNA to protein, that is the expression. So biotechnology work on these molecules which are known as genetic material. So genetic material, there are some controls. One is transcriptional control, where DNA converted into RNA. So there are some points where we can intervene or we can make changes that ultimately reflects in protein, which is the expression product that gives structural or functional units. So, and we also have some points like RNA processing control, where we can change by using various techniques, and translational controls also that ultimately gives the effect. For this, we have activators and repressors. Those are artificial or the the natural that we use for as an activator or repressor. By this, we applied on agriculture, and we get more yield. We use more nutrient from the soil to attain this the good yield. We prepare biotic and abiotic resistant plants, the herbicide resistant plants, to achieve our goal. Now, how we can improve yield? There are number of traits are going on. There are number of traits which we can target, because living system is very complex, and we have to make some changes at specific point that reflects as a product. So in this plants, the photosynthesis is the main factor, where we uh, uh, just think that CO2 plus water that give us the this sugar. So the photosynthesis, that pathway, is the main target that ultimately affects the yield, that changes shoot to root biomass, inflorescence architecture, stomatal movements, and various nutrient use efficiency, and resistant to plants. So in this Calvin cycle, there is a main thing is rubisco. Rubisco is an enzyme which help to utilize the molecular oxygen and produce sugar, that is an energy source for any work. So it has two type of activities. One is that carboxylation and oxygenation. If it joins with CO2, then ultimately this Calvin cycle going towards this carbohydrate production. But if we it has tendency to it has tendency to join with O2 also. That shifts its role towards photorespiration, that ultimately decline the yield potential. So by engineering this Rubisco, by optimizing its activity, we can change the its ultimately affect and increase the growth and yield. So by improving the Rubisco activity, by enhancing the carboxylation capacity of the enzyme, we can increase the growth and yield. So there is one example of engineering Rubisco activates from it's a thermophilic cyanobacteria in tobacco and rice, which gave the promising result in enhancing crop yield by improving photosynthesis under elevated temperature, because temperature is a major factor for growth. So another approach was also done to increase the Rubisco carboxylase activity and to decrease this oxygenase activity, so that we can shift toward the increased toward the growth and yield. The next is nutrient use efficiency. Nutrient use efficiency is defined as the production output relative to inputs, or in terms of recovery of input nutrients. That is the nutrient which are available in soil can take up by the plants and use for building their and for use for their growth and development. For example, there is a nitrogen, which is the very crucial factor for the growth. So where we can in that term, NUE, nitrogen use efficiency, we can, in terms of nitrogen, we can define it as the yield of grain relative to amount of nitrogen available to the crop from all the sources, either using fertilizer or the mineralization of organic matter in the soil or the atmospheric deposition. So one approach to improving NUE using biotechnology is to modulate plant nutrient absorption and allocation. So for this, yeah. So for this, the genetic modulation of these key controlling limited steps, like these are the enzymes, the red one, the other enzyme like nitrate reductase, nitrite reductase, the glutamine synthase, GOGAT. These all are the key enzymes responsible for accumulation of nitrogen uh, that ultimately produces the amino acid and proteins. The This nitrogen is the base for protein synthesis, and nucleotide synthesis is highly required. So any changes in these enzymes, we can uh, uh, increase the nutrient uptake and its utilization. Yes. Next is biotic and abiotic stress resistance. In biotic resistance, we can say the resistance against insects and viruses. As we all know, there is a best example of BT cotton. And for viruses, as we all experienced 2 to 3 years, there was stunted dwarf disease in rice. That is due to the virus. And there are a lot of abiotic stresses like high temperature, elevated temperature, or drought, or flood, means water scarcity or excess of water. These all are the abiotic stresses that imposed on the this plants due to the climate change, and ultimately reduces the yield in this era. So we introduce some biotechnological tools. uh, we approached to use this and produce the resistant plants. So there is a very first example, as I told you about some insect resistant plant. There are all you are aware of BT cotton, where genetically, this is a genetically modified, this is was GM crop, genetically modified organism also, we can heard about this. So what they did, this the desired DNA has been extracted from some uh, from source and cloned in plasmid, and after that plasmid it get introduced in plant in discs, or by particle bombardment method, by needle method. A number of methods are there to introduce that gene in specific cells. After that, the cells were screened under pressure, or either by antibiotic resistant, sorry, by imposing different antibiotics or various other. And after screening, the after a lot of screening, or 5 to 6 generations, when the GM plants become, then they are transplanted in fields, and the GM plants are mostly resistant to the specific insect or pest. So there is a BT cotton, as I told you. So what was the mechanism of action behind that? There is a cry protein. Cry is crystal protein produced inside the plant when a larva, this an insect bites on that. The cry protein also get accumulated inside his body and when the this cry protein get in touch with the saliva or in gut pH, it get activated into toxin and produce a crystal like structure, and that crystal like structure binds to a specific binder and increase the and produce the pore in the gut that ultimately increases the permeability, or the membrane in the cell membrane, its losses function, and ultimately insect death happens. That is the main mechanism of this wheat cotton. So there are number of target crops have been are the GM crops against the target insects, where a number of trans genes like cry1, A2, B3. There are a number of cry genes have been introduced. Next is herbicide resistance in plants. There is a EPSP EPSPS enzyme which is involved in shikimate pathway that are responsible for aromatic amino acid synthesis. So glyphosate is a herbicide. When it get in contact with this glyphosate, then by competitive inhibition, they they don't produce that particular amino acid. So ultimately, plant begins to die. But the specific herbicide resistant plants, where the transgenic plants, this plasmid which contain glyphosate resistant genes, they they are resistant to specific glyphosate, that herbicide, and ultimately the plant remains active and well flourished. The next is this. This is the very good example of starch production in Amphora potato. Starch is the main component in is a storage form of polysaccharide in potatoes. So Amphora potato, there is a it is a very good variety which took approx a long time to for the production of this. So starch is mainly in the form of amylose and amylopectin. But we desired amylopectin for some purpose, like for industry. And what happened in Amphora, this antisense gene has been introduced against normal gene. So there was no translation and no enzyme was active, so no amylose. So the ultimately Amphora potatoes have more amylopectin than the normal potatoes, which is very good characteristic or desirable trait for paper industry. So that's why Amphora potatoes were produced. So this is the small figure represents the area under GM crops, that is a genetically modified crops for various purposes, like for insect resistance, for herbicide resistance, for like this potato, Amphora, etc. So India also have 11.6 million hectare area under GM crops. Yeah, application in environment cleanness. We, as a human being, we are producing a lot of pollution by in everywhere due to this motor vehicles, due to the industries. So but nature is very good. Nature already adapts produces microbes that can clean the environment. So what is the role of biotechnology here is to identify and to find which are the functional or active genes in that particular microbes or microbial population that help to degrade the pollutants. So by using this biotechnology, by using the metagenomic based studies, we identified their genes, we enhance their copy number, and we put it in that specific polluted areas, and this quench that pollution and enhance the local macro and microflora that ultimately increases the soil health and decreases the global greenhouse gas emission, global warming, and climate change effects, etc. So now the application of biotechnology in livestock. There are a number of applications. Very first is the production of pharmaceuticals, Then donor organ production and improvement of production traits such as milk, meat, high quality of eggs, etc. Creating disease resistant animal, as we have created this insect resistance or abiotic bacterial factor resistant. Um, so animals are also disease resistant. So the very first is production of pharmaceuticals. And so biofarming is the production and use of transgenic plants and animals that is genetically engineered to produce pharmaceutical substances for the use of human and animals. This is known as molecular farming. So what happened in this? The specific DNA get, because all we know biotechnology is working on expression. That is, it work from DNA to protein. All we what we are doing here, DNA get isolated from the specific sample, restricted digested, plasmid get produced, and then after particular screening, that particular protein get harvested in large amount that is used in pharmaceutical industries. Next, the role in reproductive biotechnology. The very first is artificial insemination. All we know about the artificial insemination in animals, because there is there is a very difficult task to uh, for a farmer to sustain superior male at the farm. At every For every farmer, it's not possible. So by using this artificial insemination, that is AI, farmer can breed a number of female using the sperm from a single superior male. Either it's present 1,000 miles away, but it's a very good technique so that we can produce good quality of the new breed. It also lowers the cost of farm, because on-site this superior male or breeder male production is very difficult. Its feed or its house work, it's a lot. There is also a advantage of this AI in animals, that it by avoiding direct contact between animals, and we can stop the spread of many reproductive diseases. So the next is MOET, that is a multiple ovulation and embryo transfer technique. What happened in this? Their polyovulation has been done with the help of FSH, artificial hormones, and then artificial insemination. A number of this eggs get fertilized. Then in pregnant cow, the fertilized eggs get harvested after 7 days or after 1 week approx. Then that this embryo recovery, they get cryopreserved, or it's directly give to the receptor cow uh, for uh, for the production of a new calf or new baby. So it's a very good technique, and this to get the superior offspring or superior breed from the donor cow. Next is embryo splitting. Embryo splitting, it's an early stage reproductive technique because it creates genetically identical twins uh, from early from a single early embryo. It happens naturally, but in this IVF, in vitro fertilization, for backup, generally the embryo splitting has been introduced, so that if one get miscarriage, that we have the cryopreserved the next one for the further uh, implantation. Next is embryo cloning. Embryo cloning is the production of clone, the similar clone. It's a very simple reproductive technology where this differentiated cells taken from the organism and get inserted into denucleated this egg cell, and then egg cell get implanted into the this new female. That female gives the birth to the clone of herself. So the other biotechnological roles are the production of diagnostic immunoassays. The cell culture techniques are there, virus culture techniques are there, antibiotic production against this, monoclonal and polyclonal antibody production. So we can say the biotechnology, it's a bridge, that is a bridge between the traditional or the conventional knowledge and the modern technology, and the health of all living system that make it essential for sustainable development in agriculture and allied sciences. That biotechnology, it's a very good choice to create new plants or animals with useful traits. Okay, thank you so much.
Thank you, ma'am. On behalf of Edu-Mentors Pvt Limited, we extend our heartfelt gratitude to our esteemed speaker, Dr. Inderpal Kaur, for delivering an insightful and highly informative lecture on role of biotechnology in creating sustainable agriculture and livestock. Your inspiring lecture provided a comprehensive understanding of how biotechnology is transforming agriculture and livestock production through innovation, sustainable, and science-driven approaches. The valuable insights you shared us on the application of biotechnology in crop improvement, livestock enhancement, disease management, genetic advancement, and sustainable resource utilization we highly appreciated by all participants. Your discussion highlighted the immense potential of biotechnology in addressing global challenges such as food security, climate change, environmental sustainability, and increasing agriculture productivity, while ensuring the efficient use of natural resources. The practical examples and research based knowledge you shared have inspired students, researchers, academicians, and agriculture professionals to embrace modern biotechnological innovations for a more resilient agriculture future. We sincerely appreciate your valuable time, dedication, and willingness to share your expertise during the International Summer School Training Program on Climate Resilient Agriculture and Allied Sciences. Your contribution has made today's session highly enriching, engaging, and impactful. It has been a privilege to have you with us, and we look forward to welcoming you again for future lectures, training programs, webinars, and academic collaborations. Thank you, ma'am.
Thank you so much. Thank you so much.
Participants, you may leave now.